Hi, and welcome to Chapter 7, Part 4. In this part, we're going to talk about oxidative phosphorylation, and that's composed of two subsets, electron transport and chemiosmosis. Remember that so far, we've only been able to make 4 ATP molecules from glycolysis in the citric acid cycle, and that's been done by substrate level phosphorylation, right? Taking those phosphate groups off of other molecules. The vast majority of energy that's harvested from glucose is actually harvested from NADH and FADH2. Those are going to really account for the vast majority of energy. So those two molecules are electron carriers, and they are going to donate their electrons to the electron transport chain. And it's the electron transport chain which powers ATP synthesis through the process of oxidative phosphorylation. So let's learn a little bit more about our electron transport as we kind of remind you, I've shown you this picture before, that electron transport chain is a stepwise process. And so, and it's located in the inner membrane of the mitochondria in eukaryotic cells. If you're a prokaryotic cell, this electron transport chain is going to be located in the plasma membrane. Most of the chain's components are very complex proteins with electron carriers, and they are numbered 1 through 4 in Roman numerals. Most of the proteins are called cytochromes, and they are attached to heme groups, which function as electron carriers. Heme, that's like hemoglobin, right? That's also something that contains iron. So again, the electron carriers are going to be carrying those electrons literally between reduced states and oxidized states as they accept and donate electrons. Remember our concept of oil rig. Oxidation is loss and reduction is gain. So electrons are going to, when they're being passed through the electron transport chain, they're going to drop in their amount of free energy that's available every single time the transfer occurs. And the electrons are eventually going to join up with oxygen. Breaking the free energy drop into these small stepwise reactions does allow us to capture their energy in manageable amounts instead of a large explosion, which might happen in a chemistry class. We're having a small stepwise process in the process of making ATP. So with the electron transport chain, we are going to have a final product of water. And that's going to happen when the electrons, which are, again, for now, are being kept in NADH and FADH2, are going to be passed down this electron transport chain from one place to another, these various cytochromes. And in the end, they're going to join with water, join to form water with oxygen. Oxygen and hydrogen are going to form that water. So let's take a closer look at our oxidative phosphorylation. Again, it's an electron transport chain, which is the first thing that's happening. This is happening at the membrane between the inner membrane here, and all of these proteins are embedded in that inner membrane. So our NADH is going to join up to one of these proteins in the electron transport chain, and it's going to pass the electron down, down, down. And I'm saying down because we're changing the amount of free energy available. So energy is being released slowly and slowly. We can take a look at a little bit closer look here, kind of zoom in a bit, and notice that NADH is going to join here at the first complex of proteins, and FADH2 is going to drop its electrons off in the second. So this is called cytochrome Q. And So we get a little bit less energy from FADH2 than we do from NADH, just because the amount of free energy is less. It's a little bit higher here, it's a little bit lower here. So we make a little bit less ATP if electrons have been carried by FADH2. But as it goes along, slowly lowering in the amount of free energy that's available, eventually that electron is going to be released into the matrix. When that happens, when that electron is released into the matrix, it's going to join up with oxygen. And that oxygen is going to accept those electrons as well as some protons and make water as the byproduct. Eventually, all the result of the moving of those electrons, besides being passed to oxygen to create water, is what a big thing is happening, which I kind of skipped over up here because it's not shown in this process. And that is that a whole bunch of protons are being put into the space between the two membranes. So when we get into this idea of chemiosmosis, where we're really making tons of ATP, it is a direct result of that electron transport chain. The electrons are releasing energy as they're being passed along the electron transport chain, and it's using that energy not to directly make ATP. No, it's a very indirect process. So it's using that energy of moving those electrons down its free energy levels by pushing protons into the intermembrane space. And you see them right here. These are all protons. So those protons are pushed into the intermembrane space by the movement of those electrons, and they are allowed to move through this structure here called ATP synthase. We've established a gradient, lots and lots of protons here, not many protons here. We've used energy to do that in the form of transfer of electrons in redox reactions. And it's all pretty complicated. So we've got this gradient, and the protons are going to be allowed to diffuse down their concentration gradient. And when that happens, it releases a lot of energy, and that's what makes the ATP. Again, the name of this structure that the protons are allowed to diffuse down is called ATP synthase. And this is a release of a lot of energy, so we can use the word exergonic to describe that. And that exergonic flow is going to drive phosphorylation of ATP. This is chemiosmosis. Chemiosmosis is kind of defined as the use of proton energy in a gradient to drive cellular work. We can also use the term proton motive force to describe chemiosmosis, because its protons are moving along their concentration gradient using that to drive the synthesis of ATP. Now, again, all of this is very complicated and all very interconnected. So if you have time, I do suggest that you watch two animations that your book offers. One is the BioFlix animation of ATP synthase, and the other is an animation of electron transport. Both of these are going to be very, very helpful for you in your final understanding of, in your final understanding of how this complicated process works. So, let's take another quick look at electron transport chain and how that's connected to ATP synthesis as we put it all together, right? So here's the intermembrane space up here. And this is the inner membrane of the mitochondria, and this is the mitochondrial matrix. This section right here, this is describing the electron transport chain. And this section right here, this is chemiosmosis, which stars, of course, our lovely ATP synthase. Very important. So as electrons are passed down through the series of proteins in the electron transport chain, they are going to pump protons up into the intermembrane space. These transmembrane proteins are the ones that are doing the pumping. So we're pumping these protons against our concentration gradient, and very, very concentrated in this matrix. Also, protons are being used up in the mitochondrial matrix when electrons join up with oxygen to form water. So there's very, very little amount of protons in the matrix, and they're just going to flow right down their concentration gradient in chemiosmosis. This is incredibly important because that's where all the ATP is being made. Just a close-up here of the electron transport chain, as well as a close-up here of the ATP synthase and the process of chemiosmosis. If we do some math, we can start to account for everything that's happened. So during cellular respiration, most of the energy is going to flow in this sequence. Glucose is making NADH, and NADH is entering the electron transport chain in the form of electrons. The electron transport chain pumps all those protons into the space, and the proton motive force is what forms the ATP. About 34% of the energy in glucose molecule is transferred to ATP. That's not much, but it's still a lot. The rest is going to be lost as heat. And heat's not a bad thing. It helps us maintain our body temperature and homeostasis, but it's really 34%. We get about 32 ATPs out of a single glucose molecule. We don't actually know the exact number of ATP molecules because phosphorylation isn't directly coupled to the redox reactions. So it depends on, ATP could be, sorry, let me just back up here again and say that NAD plus and FAD, those could be made in different numbers depending on the situation. So we're just memorizing the basics of the process and keep those numbers in mind, but it does vary. Also, the proton motive force generated by the electron transport chain is used for other kinds of work. So if some of the other work needs to be done, then we won't make as many ATPs. This is just another reminder that there are some excellent animations in your book, and here's the different factors that affect ATP yield. So please go ahead and watch that animation. It's very interesting. It'll talk about what happens if there's more or less oxygen, what happens if cyanide is taken, and what happens if you uncouple some proteins. There's also an overall ATP yield animation for you to watch. So feel free to watch any of these animations from your textbook. Here's the basics though. And this is just a summary of everything that we've learned so far in a little bit less detail, but keeping track of things. So glycolysis, 2 ATPs by substrate level phosphorylation, 2 NADHs. Then we make pyruvate, and pyruvate is oxidized. We make 2 acetyl-CoAs, and we make 2 NADHs. We're also releasing some carbon dioxide here. Those 2 NADHs there could be changed into 2 FADHs depending on the situation. These 2 NADHs, though, do move into over-oxidative phosphorylation. In the citric acid cycle, we have 6 NADHs and 2 FADH2s, and those are all going to move into the oxidative phosphorylation process. So here, somewhere between 26 and 28 ATP are made. Here 2, here 2. So that adds up somewhere between 30 and 32 ATPs. Just a real quick here, because we hadn't really talked about electron shuttles, and so all I'm going to say with regard to this is that NADH could be made in glycolysis, or it could be changed to FADH2, and I think that that's probably enough for our understanding. Again, with pyruvate oxidation, 2 NADHs and 2 acetyl-CoAs, and those acetyl-CoAs go into the citric acid cycle, where we make 6 NADHs, 2 FADH2s, and 2 ATPs through substrate level phosphorylation. And finally, we take those NADHs and FADH2s, run them through the electron transport chain and chemiosmosis to get our final product. That's a lot to learn. We're not quite done because we have to learn a little bit about fermentation and maybe a little bit about anaerobic respiration. So stand by as we get to learn Chapter 7, Part 5.